logistic relative risk increase. But the association between alcohol consumption and the development of chronic pancreatitis for sure is there. But there's also some little hope. If you take a look at this picture, then you can see that the risk to develop chronic pancreatitis is much less than the risk to develop liver cirrhosis. So all of us have the chance before we develop chronic pancreatitis that we maybe get alcoholic liver cirrhosis if we drink too much. And also for this kind of disease, there seems to be some threshold that you need to drink at least a certain amount of alcohol before you are able to develop the disease. And if you take a look at the data, then it seems rather implicative that there might be a genetic basis for both kind of disease. And let's see if there are some genetic studies that help us to understand the pathogenesis of alcoholic chronic pancreatitis. Before we hand over to this topic, I want to show you just briefly something. If you take a look at the disease course of both kinds of disease, which means that you compare patients that rather have idiopathic or hereditary chronic pancreatitis compared to patients with alcoholic chronic pancreatitis, it might be that you would think that these patients that have the inherited disease, which starts quite early, compared to the patients with alcohol-related disease, that they might have a much more severe disease course. But if you take a look at the data published by Volker Keim from our clinic, then you can see that if you take a look at calcifications here on the left, that the alcoholics have much more calcifications compared to PRSS1 associated chronic pancreatitis patient or idiopathic chronic pancreatitis patients. And it seems to be the same for diabetes, that these patients that have alcohol-related disease have with a much higher probability the development of diabetes over the time course compared to the patients with hereditary or idiopathic chronic pancreatitis. So there seem to be some factors that influence this disease in addition to the alcohol consumption. And we have heard a lot of data from Dave about smoking and so on. So when we talk about genetics in alcoholic chronic pancreatitis, it's quite easy. There's not that much that you can show about associations with this kind of disease. It is proven that there are SPINK1 mutations that you will find more common in patients with alcohol-related disease compared to normal controls. Of course, if you want to be quite sure that this is not an effect that you measure, then you might even compare these data to patients that have a comparable alcohol consumption but no overt chronic pancreatitis. But I guess if you would do this in addition, which hasn't been done in this publication, then this association would also be there and would not vanish. Another thing is chymotrypsin C, which is also associated with alcohol-related chronic pancreatitis. And in this gene, this is one variant that was associated in this publication from our clinic together with Miklos. You can see the R254W variant seems to increase the risk to develop alcoholic chronic pancreatitis with 5.1. So this is quite a huge odds ratio for alcohol-related chronic pancreatitis, as you will see later on, and maybe it's a little bit lower if we expand the analysis to larger data sets. Of course, time goes on, and this is one of the publications we have talked about a little here, and I want to show you some more details of this publication because this is a very important next step if you perform genetic studies in chronic pancreatitis, that you move on to genome-wide association studies. You don't have to be too sad if you take a look at the odds ratios that come up with these studies because they are much lower, but this is just due to the method that is used when you perform genome-wide association studies. And this study was able to identify variants, common variants in different loci, which is, for example, the Claudin-2 locus and the PRSS1 locus that associate with alcohol-related chronic pancreatitis and in addition with non-alcohol-related chronic pancreatitis. And these are the most important SNPs that have been identified by this large approach. And you can see the one SNP which is associated is located next to the PRSS1 gene. And we can see here, if you take a look at the data, it's enough if you take a look at the conjoined analysis here on the right, that this is a protective variant. We have an odds ratio below 1, so this variant with a significant genome-wide p-value protects against the development of chronic pancreatitis in this cohort. And then there are other variants, and this is quite interesting because now we have variants that are located on the X chromosome. So this is the first time that there is an association to variants that might have a different effect in different genders. So what you can see here, the first variant located on the X chromosome, this time it's a positive odds ratio, so this is a risk contributor to chronic pancreatitis, and the genome-wide significance was reached. And the same holds true for the other variant located on the X chromosome. It's also in risk increase, and we have an odds ratio of around 1.4, and genome-wide significance is for sure reached in the conjoined analysis of the data set. So before you make the effort to put all these data you have into a construct which might come to the clinical use, then you have to replicate genetic findings. That is always important to show if you can find the same results in other cohorts. And so this is what we did. We analyzed a large European cohort of patients with alcohol-related chronic pancreatitis and idiopathic or non-alcohol-related chronic pancreatitis for these three associated variants. And this is the results for the PRSS1 variants we found in our cohort. What you can see on this slide, it's quite busy. I learned this from Heiko, he's my mentor, so he showed me how to make busy slides, is that you have an NACP non-alcohol related cohort here on the top of this graph, and you can see that we have a significant p-value here, 0.04, and then we have a large number of samples and controls investigated, and you can see that this variant contributes risk or protection against chronic pancreatitis with an odds ratio of 0.9 in our cohort. In addition, we have for sure replicated this in patients with alcohol-related disease, and this is the graph shown here below. And once again, a large cohort of patients throughout Europe with alcohol-related disease and a large number of controls. The p-value is significant, and you can see that this variant protects against alcoholic chronic pancreatitis. Odds ratio is 0.6 around. What you now have to show for sure is that this variant is not an alcohol dependence locus. So we compared this data to patients with alcoholic liver cirrhosis that don't have chronic pancreatitis. And in addition, we compared this data to patients from Germany with alcohol dependence with no chronic pancreatitis. And what you can see in this groups is that the association is still there. You can see the odds ratio is quite the same. And the p-values are highly significant, and I guess the number, these are only German patients, is sufficient that you can really now conclude that these variants do not contribute to alcohol dependence. They are real variants that contribute to the pathogenesis of alcoholic chronic pancreatitis. So we can conclude that this PRSS1 SNP seems to protect a little bit more against alcohol-related disease than non-alcohol-related disease, or maybe it's better formulated that you can say the effect is greater. It's more protection against alcohol-related disease. Of course, there are these other SNPs on the X chromosome, and this is quite difficult to analyze. You can see that at least genders carry different amounts of X chromosomes, so you have to be careful with the analysis on the X chromosome. And what you can find out here in the group of ACP males is that the association for this Claudin 2 variant is there. You can see once again a significant p-value. The number of male ACP patients is expectedly high and sufficient to address this question, and the odds ratio is somewhere 1.6. And once again, the comparison against patients with alcoholic liver cirrhosis or alcohol dependence, and also in this cohorts, the association is there. So also this variant in male patients is no alcohol dependent variant, and it is a variant that contributes to the risk in alcoholic chronic pancreatitis. In females, of course, it's a little bit more difficult because there are not many patients in the ACP cohorts that are females, actually, but this maybe will change over the years. And you can see the numbers are rather low here, but we can confirm that also in females, this Claudin 2 variant contributes to a risk increase in alcohol-related chronic pancreatitis. And once again, it's not an alcohol-dependent locus, so it's really a variant that also influences the risk to develop chronic pancreatitis. And we did the same for the other variant, but I'm not going to show you these data in addition. So what can we conclude so far for the ACP part of the talk? We can say for sure that it seems that there's a difference between the disease in patients with alcohol-related disease. Maybe we can say the disease progresses a little bit faster, but there are several explanations why this could be the cause. Maybe it's even more severe. That's what we learn in our daily clinical practice, that many patients have to be, for example, operated on because of constant pain in these groups with alcohol-dependent chronic pancreatitis. There is a genetic association in alcoholic chronic pancreatitis, but it's less pronounced than we have learned for non-alcohol-related disease. We have only the SPINK1N34S variant associated with the disease, and in addition, the CTRCR254W variant. And the other associations, tomorrow Markus Lörch will present you some data, are rather weak. So then we have the genome-wide association study, and we are all happy that this study contributed to a new pathway in the development of chronic pancreatitis. This will initiate a lot of research, and maybe we will learn a lot of more things about how chronic pancreatitis can develop in the next year. So these variants contribute to NICP and ACP disease development, and it is very important to stress that these variants are not variants that are associated with alcohol dependence. They are clear variants that contribute to chronic pancreatitis risk. So now we make a kind of transition from alcohol-related chronic pancreatitis to non-alcohol-related chronic pancreatitis and back. And I want to show you some interesting data about carboxyl ester lipase. It's a major component of the pancreatic juice. You all know this. And it's responsible for the hydrolysis of cholesterol as well as for other esters. And as you have read in the publications, there is a disease which is associated with a disturbance of this gene. It's MODY8. It's a monogenic syndrome, which leads to endocrine and exocrine dysfunction of the pancreas. And so it was an idea to investigate this gene in chronic pancreatitis patients. And this idea didn't came up from our groups. It was a Norwegian group that came up with this idea. So this is CEL. The interesting thing when you take a look at CEL is that there is CEL here in the front, 11 exons, and then we have a pseudogene, CELP, which is behind the CEL gene, which is quite similar, but you see that the exons 2 to 7 are lacking in the pseudogene. And there's one interesting thing you already have heard of. There's a VNTR, so we have a variable number of tandem repeats in the exon 11. And these consist of 33 base pairs, and the most common variant that you can detect in your patients will be 16 repeats. And there have been some investigation of this VNTR in alcoholic chronic pancreatitis patients, and I want to show you briefly the data before we move on to the more interesting aspect of the CEL gene. This is one possibility to screen this VNTR. It's quite difficult to do this analysis by other methods. It's not the best method to analyze such VNTRs. You can make this a little bit more appropriate with different methods. But what you can see here in this publication by a Japanese group from 2005 is that you have different genotypes. You can have a normal genotype, a small, so a smaller number of VNTR compared together with the normal alleles. Then you have normal and large, and small and large, and all the other possibilities that you can detect. And this was the method this group applied to analyze this VNTR region in patients with chronic pancreatitis. And what they found out is, in this quite busy slide again, that there is an association of these combination of the normal allele with the long allele, which is the NL genotype, with alcoholic chronic pancreatitis. And you can see they investigated around 328 male control subjects and around 100 male alcoholic chronic pancreatitis patients. And then in the end, if you compare this NL genotype between the groups, there's a difference, which is around 28 to 39%. Here is the 39% and here 28%. And this leads to a significant p-value with an odds ratio, once again low, of 1.8. And so the authors concluded that this VNTR variant contributes to the pathogenesis of alcoholic chronic pancreatitis. As stated before, what you need when you perform such studies is replication. So there is another work. This is from the Norwegian group. I will show you more data from this group with the next slides. And they analyzed these VNTR again in some patients with alcohol-related disease and idiopathic disease. You can see 203 patients with alcohol chronic pancreatitis and 64 patients with idiopathic chronic pancreatitis and around 400 controls. And if you look at their data, then you can see there is no association. So it seems that the formally described association of the Japanese group might be some chance finding, but of course, if you take a look at the numbers, it's just 203 patients and only 64 idiopathic patients. It seems that we need a larger data set in that these VNTR is analyzed before we really conclude that there is no association with alcoholic chronic pancreatitis of this VNTR. So once again, back to the CEL. Here, what you can see is an interesting thing that happened during the evolution of this gene, and what really happened in the Norwegian were able to identify this is that there's homologous recombination, and we have two different alleles that result of this homologous recombination. We have once a duplicated hybrid allele, so there's part of the pseudogene inserted here in between the normal CEL gene, so it's a larger product. And then we have a deleted hybrid allele where the exons 10 and 11, 10 in parts, are merged with the normal cell hybrid, with a normal cell gene, and so we have a deleted hybrid allele. And they have done a lot of work, so I'm just showing you the essentials of these publications to investigate this hybrid allele, and it seems that the deleted hybrid allele somehow confers a risk to chronic pancreatitis. This is another image of the hybrid allele, so to better understand what might happen on a functional level thereafter, you can see that there are bile salt and binding sites which are not affected in the deleted hybrid allele. There's no change after the deletion. The VNTR is shorter by the deletion, and so it might be that the glycosylation sites are missing, and this might, for example, explain the functional consequences of the deleted hybrid allele. So there are different ways to investigate this hybrid allele. First of all, the complete region of the CEL gene was investigated, so there were no variants that other than the hybrid allele associated with chronic pancreatitis. This was the first step undertaken in the study. And then in the end, it turned out that this hybrid allele might be a risk factor, and so a light cycler assay was designed by Heiko Witt to investigate this gene, which is quite good because you can really fastly investigate a large set of samples. And what you can see here in this melting curve assay is that the CEL deleted hybrid allele has a higher melting point than the wild type here. And if you show it like this, then you can quite easily see that there are differences between these wild type patients and the patients carrying the deleted hybrid allele. The only thing which is not perfect in this assay is that this assay is able to detect false positive samples. So you need another method to really show that all these samples that are positive here are real positives, and therefore another assay was invented, and this is a PCR, long PCR range assay, and then a gel analysis, which is quite time consuming, which is also able to detect the deleted hybrid allele. You can see here that the primers are so designed that you can cover both the long or wild type situation and then the deleted situation. And if you take a look at the gel, then you can see the wild type is here. And if you have the deleted allele, then you have a smaller band down here and the normal band here. So this is heterozygous patients for the deleted hybrid allele. And this method allows you quite easily to identify the hybrid allele carriers in the patients and controls. So this was done in quite a lot of samples, as you can see here. There was a screening cohort that helped to enforce all the other investigations thereafter. We have here a screening cohort from Greifswald. 10 of 71 patients carried this hybrid variant. So we have an odds ratio of 15.5 in this group. But of course, this is a small number of samples, so you have to wait for the other groups. And when you take a look at the replication cohorts from Munich, Leipzig, and from France, then you can see that in each of the groups, there is an overrepresentation of the deleted hybrid allele in the patients compared to the controls. And if you combine this in a kind of formal meta-analysis, then you can see that the odds ratio for this deleted hybrid allele is 6.4, and it's a highly significant p-value. So this seems to be quite sure a new pancreatitis gene, which is in the range of CTRC concerning the odds ratio. And which is quite elegant, if you have the chance to do it, then we also investigated patients with alcohol-related chronic pancreatitis, and you can see there's also an association in this group. This effect is much lower. You can see only 1.8% of the patients carry this variant compared to 0.8% in the controls, but this is still significant with an odds ratio of 2.3. So we have also another alcohol chronic pancreatitis gene that is displayed here. Of course, it's always quite good if you have, in addition, some functional data that support your work, and it's not only genetics. And the Norwegian group have done a lot of efforts to show what happens on a functional level, how this deleted hybrid allele confers risk to chronic pancreatitis. And I just show one picture because Miklos will ask a lot of questions about the functionality. And what they show here is that if you take a look at the specific enzyme activity of the CEL gene and the deleted hybrid allele, and then you can see that it has impaired activity. But this is not the only mechanism that explains how this might contribute on a function level to chronic pancreatitis. There seems to be retention within the cells, and this might lead to autophagy. There are some quite good data that showed it. And
so how does this explain pancreatitis?
Yeah, it's the same with CPA1.
Same mechanism.
Yeah. We have just to believe it. Maybe the most important thing. At least the genetics are quite convincing. And so there seems to be a new pancreatitis gene which might be needed to test also in other groups in the future. So if we sum this up, it seems that there have been some progress in the last years. Heiko published after 8, 9, 10, maybe even more years, the CPA1 story. GWAS, and this is quite important, can be successful. And I hope that our data on alcoholic chronic pancreatitis, our GWAS data will come up soon, and we might even show other pathways. Then there is an influence of a potential influence of this VNTR region of the CEL gene, but we need a larger study to really know if it's true or not. And it seems that this deleted hybrid allele confers risk more to non-alcohol-related chronic pancreatitis than to alcohol-related chronic pancreatitis, but at least the genetic data is are quite good and convincing, and the functional data, it might be impaired activity, autophagy, we will see. So far, the reviewers are quite okay with the explanations presented in the submitted paper. Okay, thank you very much.
So I have two questions. First one's very simple. So you actually replicated the Japanese finding?
No, we didn't do anything so far with the VNTR, but I think it's needed. There's one effort undertaken by the Liverpool group, and they show that there might be something, but the data set of them is quite small.
Does it look like they had a smaller protein size, which may correspond with what you found? Is that correct?
Concerning the VNTR?
Yeah.
On a functional level, no.
Okay. The other question was that since the variants in the PRSS1, PRSS2 locus that result in diminished expression of trypsin are protective against alcoholic pancreatitis, does that mean that alcoholic pancreatitis is associated with a trypsin-driven pathology?
Yes. Exactly.
Now I'm in between seats. I have to say yes, because if I say no, then I have a problem with Miklos. But
Say what you mean. And mean what you say.
It's quite difficult. Of course, maybe there is some influence of trypsin activity also in alcohol-related disease, but the effect is low. So maybe this doesn't change.
But this is my question. If I look at your data set and Dave's data set, the effect of the PRSS1 variant is stronger in the alcoholic population.
Yeah. That's true. Yeah.
Which is totally counterintuitive, right?
Yes, I know.
It looks like idiopathic is not driven by trypsin, but alcoholic is, in both studies.
But maybe the functional effect of this variant is rather low. So, but I don't know. I have no good explanation. I have just to wait maybe 10, 20 years until you forget to ask this question again. And then.
So you showed in one of the earlier slides that alcoholic patients have more calcifications than their pancreas, and this could be because I'm a pediatric gastroenterologist. I don't see alcohol much. So why is that exactly? So why does alcohol cause calcifications? Is that a prognostic?
I have not the best explanation for it.
The calcifications are probably due to smoking.
That's for sure true. At least a possibility, because all of...
Because alcoholics smoke.
Why does smoking cause cancer?
Yeah.
I don't know what, but that's
Because of
common,
the alcohol.
common observation.
But
Congratulations, John,
Thanks.
for the fantastic data with a very important clinical implications. And I'm very flattered that you did allow us to contribute a little bit to one of your studies. We have a very big experience in Poland with alcoholic patients, and I was always wondering how come that they either get acute pancreatitis or chronic pancreatitis, either alcoholic liver disease, how it happens together. And I wonder if you have any genetic explanation for that, because is it like alcoholic which have liver disease, they do not have this mutation that contribute to acute pancreatitis, or do they have some other, by some chance, maybe you know, maybe they have some other mutation that contribute in particular to liver disease? What do you think?
it's a perfect question, and this question, since I work in Leipzig with Joachim Mössner together, from the first day he asked us this question. He wants us to resolve this problem, but nobody really has resolved it so far. So what we have from the data, there's now underway a genome-wide association study in alcoholic liver cirrhosis. There seem to be two hits, and they can compare this data to our alcoholic chronic pancreatitis patients, but this work is not done to see whether there is something that correlates with each other. Yeah. And the other thing is, for sure, there are at least, let's say, 10, 20% of patients with both kind of disease, if you take a look at autopsy studies. So I think in the end it will be very, very difficult to find a real good explanation why the one goes into this direction and the other into that.
The very last.
Just to answer the question, what alcohol and smoking do is drive fibrosis. You have to have an injury in a tissue to initiate the process. And if you have mutations like trypsin mutations or others that target the pancreas, and you drink and smoke, it drives chronic pancreatitis. If you have an injury in the liver, such as hepatitis C or others, and you drink and smoke, it drives the fibrosis. So that the drinking and smoking modifies the immune response to drive fibrosis, and the organ that you get the fibrosis in depends on where the injury is. But there's also a much greater overlap between the two than you expect by chance alone. So you see chronic pancreatitis and liver disease far more commonly together than you would have predicted, suggesting that there is an overlapping mechanism.
Yeah, but still it's, we don't observe it at such a big number. And of course I understand that the fibrosis in both diseases is vital, but still I would expect to have bigger number of the patient they have both.
Yeah, you'd expect 1 in 1,000, and we see about 10 in 100.
Yeah,
Thank you
around
for
this.
the very active